Welcome to an exploration of one of the most fascinating puzzles in the origin of life, the Droplet Division Mystery. When certain liquid droplets experience stress or chemical reactions, they spontaneously divide into smaller droplets, mimicking biological reproduction. Let's begin by understanding what these dividing droplets actually are. These are liquid droplets, suspended in another liquid medium or in air, forming what scientists call an emulsion. Under specific physical or chemical conditions, these droplets can spontaneously split into two or more smaller droplets. Remarkably, this division requires no biological machinery whatsoever. It's driven purely by physical and chemical processes. Here's how it happens. A spherical droplet elongates under stress, forming a dumbbell shape, then splits into two separate droplets. What physical principles govern this fascinating process? Surface tension is the key force at play. It's the tendency of liquid surfaces to minimize their area. Surface tension gamma equals force F divided by length L, measured in newtons per meter. For a droplet, the surface energy is directly proportional to its surface area. Surface energy equals gamma times area, which for a sphere is gamma times 4 pi r squared. Here's an interesting question. Why would a droplet divide if division actually increases the total surface area, raising energy? The answer is that other forces must overcome surface tension. These include chemical gradients, mechanical stress, or Marangoni effects. Surface tension acts like an elastic skin, pulling the droplet inward to minimize surface area and maintain a spherical shape. One crucial mechanism is the Rayleigh plateau instability, discovered in the 1800s. Lord Rayleigh and Joseph Plateau discovered that long cylindrical fluid columns are inherently unstable. If the wavelength of a perturbation lambda exceeds 2 pi times the cylinder radius r, the instability grows. Small random perturbations in the cylinder's radius grow exponentially, causing breakup into droplets. A cylindrical thread of liquid develops small perturbations that grow until the thread breaks into a chain of droplets. The breakup time scales as density ρ times radius cubed divided by surface tension gamma. Chemical reactions can also drive droplet division through fascinating mechanisms. Surfactant molecules are chemicals that reduce surface tension locally where they accumulate. Surface tension with surfactants is less than without them, creating gradients. These surface tension gradients create Marangoni flows, where liquid moves from low to high surface tension regions. Surfactant molecules at the droplet surface create flows that push liquid outward, potentially causing elongation and division. A classic example: oil droplets in water with added soap will divide as soap molecules accumulate unevenly on the surface. Let's examine the energetics of droplet division more carefully. Consider a sphere of radius r that splits into two equal spheres. What's the energy change? The initial volume is 4/3 pi r cubed. The final volume is 2 times 4/3 pi r cubed, where r is the radius of each daughter droplet. By volume conservation, r cubed equals 2r cubed. Therefore, r equals r times 2 to the negative 1/3, which is approximately 0.794 times r. The initial surface area is 4 pi r squared. The final surface area is 8 pi r squared for both droplets combined. Substituting, the final area is approximately 5.04 pi r squared. The change in area is 1.04 pi r squared, a 26% increase. So division increases surface area by 26%, requiring energy input from somewhere. The energy cost is gamma times 1.04 pi r squared. Let's work through a concrete numerical example to make this tangible. Consider a water droplet in oil with radius 1 millimeter. R equals 1 millimeter, or 10 to the minus 3 meters. The water-oil surface tension is approximately 0.05 newtons per meter. The energy required for division is 0.05 times 1.04 pi times 10 to the minus 6. This equals approximately 1.6, 3 times 10 to the minus 7 joules. To put this in perspective, this is about 40 trillion times smaller than a single food calorie. So here's the question. Where does even this tiny amount of energy come from? The answer is that it comes from chemical reactions releasing energy, or from external mechanical work applied to the system. Now let's explore why this matters for understanding life's origin. Early life required compartmentalization, separating internal chemistry from the external environment. Cell membranes perform this function in all modern life, but how did the first membranes form? Protocells, the precursors to cells, likely had simple lipid membranes surrounding internal chemistry. If these protocells could divide like droplets, they would have a primitive form of reproduction. Fatty acid vesicles are leading candidates for primitive cell membranes. Fatty acids are simple molecules with a long hydrocarbon chain and an acidic head group. Their chemical formula is CH3, followed by CH2 repeated n times, ending in COOH. In water, fatty acids spontaneously form vesicles, hollow spheres with bilayer membranes. The vesicle has a bilayer membrane with hydrophobic tails pointing inward and hydrophilic heads facing the water. Remarkably, experiments show these fatty acid vesicles can both grow and divide under certain conditions. Let's examine the growth and division cycle of fatty acid vesicles. Step 1. When you add more fatty acids to the solution, they incorporate into existing vesicle membranes. Step 2. As fatty acids join the membrane, the vesicle surface area increases. Step 3. If the internal solution is more concentrated than outside, osmotic pressure builds up. Osmotic pressure, pi, equals concentration C times the gas constant R times temperature T. Step 4. The osmotic pressure causes the vesicle to elongate and become mechanically unstable. Step 5. Division occurs through the Rayleigh plateau instability we discussed earlier. The complete cycle. A small vesicle grows by incorporating fatty acids, elongates under osmotic pressure, then divides into two daughter vesicles. What experimental evidence supports this scenario? Jack Szostak's laboratory at Harvard has demonstrated fatty acid vesicle division in controlled experiments. Here are the key findings from their groundbreaking research. Vesicles grow when fed fatty acid micelles, small aggregates of fatty acid molecules. Osmotic swelling, caused by higher internal solute concentration, drives elongation. Gentle mechanical agitation, like shaking or fluid shear, triggers the division process. The division produces functional daughter vesicles that can themselves grow and divide again. Let's quantify some aspects of this process with real numbers. Here are typical parameters for fatty acid vesicles in experiments. Radius typically ranges from 1 to 10 micrometers. Membrane thickness is approximately 3 to 4 nanometers. Surface tension ranges from 0.001 to 0.01 newtons per meter. The growth rate depends on the concentration of fatty acids in the surrounding solution. The rate of area change is proportional to the micelle concentration of fatty acids. What are the typical timescales for these processes? Growth from small to dividing size takes minutes to hours. The actual division event happens on the timescale of seconds to minutes. For true reproduction, division must be coupled to internal chemistry. Modern cells tightly couple division to DNA replication, ensuring each daughter gets genetic material. Primitive protocells might have coupled division to RNA replication in a simpler way. A protocell containing RNA molecules that can replicate using the internal chemistry. As RNA molecules replicate, they produce more molecules inside the vesicle. More molecules means higher internal concentration, which increases osmotic pressure. Increased RNA copies leads to higher osmotic pressure, which promotes division. This creates a primitive coupling between heredity through RNA and division, a key step toward life. Let's address some critical questions about this scenario. First question, were fatty acids actually available on the early Earth? Answer, yes. Fatty acids have been found in meteorites and can be synthesized through prebiotic chemistry. Second question, how is RNA partitioned between daughter vesicles during division? Answer, it's initially random, but various mechanisms can enrich certain vesicles with more RNA. Third question, can this simple system actually lead to evolution? Answer, yes. If RNA molecules can mutate and some mutations affect the vesicle's growth or division rate. Let's explore how competition could drive early evolution. Different protocells would compete for available fatty acids in the environment. Protocells that grow faster will incorporate more fatty acids from the shared pool. Faster division means producing more offspring protocells in the same time period. Starting with equal populations, faster dividing protocells produce more offspring and dominate the population over time. Population grows as n0 times 2 to the power of time divided by division time. Natural selection for efficient growth and division emerges naturally from this competition. Let's be honest about the limitations and challenges of this model. Division in these systems is not perfectly controlled like in modern cells. RNA partitioning between daughters is random without sophisticated mechanisms. The minimum chemical complexity needed for meaningful evolution is still unclear. Environmental conditions must be just right, including pH, temperature, and ionic strength. There's a narrow optimal parameter space where conditions are just right for protocell growth and division. Are there alternative mechanisms for primitive cell division? Freezing and thawing cycles can physically fracture vesicles, creating crude division. Wet-dry cycles in tidal pools concentrate molecules and can cause vesicle budding. Interactions with mineral surfaces like clay can template and divide vesicles. Clay mineral surfaces can absorb vesicles and facilitate their division through surface interactions. Each of these alternative mechanisms has its own advantages and disadvantages for early life. What are researchers investigating today in this field? Scientists are building synthetic minimal cells that can divide, testing what's truly essential. Research focuses on coupling RNA replication chemistry to protocell growth and division. Evolution experiments with populations of dividing vesicles to see if selection can occur. Geochemists are working to understand what environments on early Earth could support protocells. The research pathway: studying protocells in the lab to understand the bridge from chemistry to life. What does this tell us about the possibility of life elsewhere in the universe? The physical principles of surface tension and droplet division are universal, working the same everywhere. Simple organic molecules, including fatty acids, are common in meteorites and interstellar space. Liquid water appears to be the key requirement, as it enables the chemistry and physics we've discussed. This suggests life might emerge wherever the right conditions exist, making it potentially common in the universe. From emergence to evolution, the journey of life can potentially unfold anywhere in the universe where conditions are right.